Usually when we talk about a major technological shift, there's this expectation of visibility. Like, if engineers build a faster car, you can physically watch it speed down the track. You write a smarter algorithm for your email, and, you instantly see less spam in your inbox. The progress is just right there in front of you.
Right. It's measurable in our daily routine. I mean,
Yeah.
we are really wired to believe that if something is revolutionary, it must be loud and obvious.
Yeah, but then you step into the world of molecular biology in July 2026, and suddenly that visibility just vanishes completely. We are looking at a technological landscape operating entirely at the microscopic level, but its implications are world-shattering. It is quite literally writing the code of life.
It really is, and it's doing it from the ground up.
And that's exactly what we are exploring today through the lens of a new comprehensive survey paper on generative AI for controllable protein sequence design. We're on a mission today to unpack how we are moving from, just hoping we stumble upon the right biological code to directly typing it out.
To appreciate how wild that shift is, you have to look at how we used to do this. For decades, if a scientist wanted a new protein to, say, break down a chemical or treat a disease, they relied on directed evolution.
Which is basically just guessing, right?
Exactly. You take a protein, mutate it randomly, and pray it gets better. You're pipetting, heating, checking in a lab over and over.
The paper actually describes the mathematical number of possible protein sequences as being greater than the atoms in the observable universe. So directed evolution sounds less like engineering and more like, finding a single specific grain of sand on a beach.
Oh, it's worse than finding a needle in a multidimensional haystack. Because in this biological haystack, if you pick the wrong sequence, it chemically degrades into useless mush in a petri dish. Directed evolution was blind, incredibly expensive trial and error. Generative AI fundamentally replaces that blindness with intent.
Okay, let's unpack this. Because before we can command an AI to build a specific microscopic machine, we first have to understand how it speaks the language of biology. The source material compares protein sequences to sentences, where the 20 standard amino acids are basically the letters of the alphabet.
Right, the building blocks.
And naturally, scientists just threw the exact same AI models that write text at these biological sentences. But here's my hang-up. A text-based AI reads and writes left to right, like a book. A protein doesn't exist as a flat line. It folds into this highly complex 3D blob. So how does a left-to-right algorithm handle 3D physical reality?
Well, that tension cuts right to the heart of the first major category of models in the survey, which are autoregressive models, or ARMs.
Okay.
Models like ProtGPT-2 or ProGen-2, which, by the way, scales all the way up to 6.4 billion parameters, they actually do generate left to right. They predict the next amino acid based purely on a sequence that came before it.
Which means they scale beautifully, right? Because we already know how to train massive language models. We do it for text all the time.
Exactly. They capture the global mathematical dependencies of sequences incredibly well. But, and this is crucial, your hang-up is totally valid.
Yeah.
Nature didn't evolve proteins by starting at the left side of a chain and guessing the next letter.
Yeah, evolution is nonlinear.
Right. So while ARMs are powerful, being forced to guess left to right is a distinct limitation when the physical goal is a 3D folded structure.
So what is the alternative to left-to-right guessing? The survey brings up variational autoencoders, or VAEs, but the math there seems completely different.
It is. Think of a VAE less like a text generator and more like a massive mixing board in a music studio. You have this incredibly complex topographic map of millions of natural proteins. A VAE uses an encoder to compress all that hypercomplex sequence data down into a few fundamental mathematical sliders. We call this compressed map a continuous latent space.
Continuous latent space. Okay, so instead of dealing with thousands of individual amino acids, I'm looking at a mixing board with a few distinct sliders.
Yeah.
If I push the, hydrophobia slider up slightly, what happens?
The AI's decoder takes that new, slightly adjusted slider position and translates it back into a full, brand new protein sequence. And because the latent space is mathematically continuous, you can smoothly navigate between different protein properties without generating total gibberish.
Wow.
You also have generative adversarial networks, or GANs, taking a slightly different approach.
Right, GANs, the forger and the critic. The generator builds a fake sequence, and the discriminator tries to spot the fake.
Exactly. And the discriminator has studied millions of natural proteins, so it's ruthless. A model called Protein GAN uses this relentless competition to expand functional sequence spaces. It allows scientists to safely generate highly diverse variants that possess physical properties completely indistinguishable from natural proteins.
But the real showstopper in the survey, I mean, the one that completely solves my left-to-right hang-up, is the diffusion model.
Oh, absolutely. What's fascinating here is that diffusion models like EvoDiff pull their inspiration directly from non-equilibrium thermodynamics. Instead of writing a sequence letter by letter, you take a perfectly valid functional protein sequence and gradually corrupt it. You inject mathematical noise into the data until the sequence becomes total static.
Like taking a high-definition photograph of a landscape and slowly adding gray static until the image is entirely gone.
Exactly like that. Then the neural network is tasked with reversing that corruption. It learns how to take pure noise and iteratively refine it back into a coherent, functioning protein sequence.
And because it's starting from a cloud of static, it's order agnostic. It's not guessing the next letter. It is looking at the whole fuzzy picture at once.
Which is exactly how it understands 3D space on a 1D chain. Because diffusion isn't reading left to right, it can look at amino acid number 10 and amino acid number 400 simultaneously.
Right.
It figures out how they mathematically attract each other in 3D physical space as the noise clears. It mirrors how a folded protein actually exists, allowing it to generate proteins with disordered regions, parts that don't have a fixed shape but are functionally critical. Left-to-right models struggle massively with that.
So just to lock this in for you listening, we have an incredible toolbox. Autoregressive models predicting the next letter, VAEs mixing properties smoothly on a latent soundboard, genes playing the forger and critic, and diffusion models unblurring the whole 3D picture at once.
It's a robust set of tools.
It is. But what stands out to me in that toolkit is that generating random functioning proteins is a cool party trick, but it's totally useless if I'm trying to cure a very specific liver disease. How do we actually force this AI to build what we want rather than just whatever it feels it's making?
Well, we enforce control by exploiting the fundamental rule of biology. Sequence determines structure, and structure determines function. Because of that unbroken causal chain, we can grab the steering wheel at different levels. Let's start with structure to sequence control, which is commonly known as inverse folding.
Hold on, I thought the massive breakthrough a few years ago was AlphaFold, which took a 1D sequence and finally predicted the 3D structure. Isn't inverse folding going completely backward?
It is backward, and that's exactly the point. In inverse folding, we already know the ideal 3D shape we need. Imagine we need a protein shaped exactly like a microscopic wrench to permanently block a cellular receptor. We hand that target structure to the AI. The AI's job is to engineer the 1D amino acid sequence that, once synthesized, will spontaneously fold itself into that exact wrench shape.
But physically, multiple different sequences could theoretically fold into that same shape, couldn't they?
They absolutely can, which is why the AI relies on specialized models to find the optimal one. The survey highlights protein MPNN, which generates the sequence autoregressively, but constantly checks its math against the structural constraints.
Got it.
There are also one-shot models like PyFold that calculate the sequence in parallel to save immense amounts of compute time, and diffusion bridge models like Bridge-IVE that iteratively tweak the sequence from both ends until the physical structure constraint is mathematically satisfied.
Okay, here's where it gets really interesting. What if we don't know the exact molecular shape we need? What if we just know the biological job we want done? That brings us to function-to-sequence constraints. The survey details how scientists can literally use textual prompts. I mean, it's like using Midjourney, but to generate artificial enzymes.
It sounds like sci-fi, but models like ZymCTRL allow you to input an enzyme commission number, which is essentially just a standardized text classification of an enzyme's biological function, as a direct prompt. You can also use a fitness score, demanding the AI optimize a sequence until its mathematical probability to survive and function in a high-temperature environment maxes out.
My favorite mechanism here is binding targets. You can show the AI a specific molecule, like the spike protein on a novel virus, and command the AI, build me a key that perfectly fits this exact lock.
That is where the sheer computational force of these generative models shines. They aren't just matching puzzle shapes. The AI is calculating the biochemical affinities, the electrical charges, the hydrophobicity. It is designing a molecular puzzle piece that inherently wants to magnetically snap onto the virus target.
So the algorithm takes our structural constraint, clears the mathematical noise, and spits out a sequence that looks flawless on a computer screen. Do we immediately take this code and spend hundreds of thousands of dollars synthesizing it in a physical wet lab?
No. Doing that would bankrupt a research lab overnight.
Yeah, I figured.
An AI's output is nothing more than a hypothesis until it survives the physics of the real world. To prevent synthesizing biological garbage, we have to employ rigorous digital bouncers. We call this in silico evaluation.
Because if the AI hallucinated, we need to catch it digitally before we waste real money.
Precisely. First, we run the sequence back through AlphaFold to check its structural confidence using a PLDDT score.
What exactly is a PLDDT score measuring? The survey mentions it, but how does it prove the protein won't just fall apart?
It is measuring the model's confidence in the physical distances between local atoms. If AlphaFold is highly confident that these specific carbon and nitrogen atoms will sit exactly where it predicts based on physical laws, the sequence gets a high PLDDT score. It passes the first bouncer. Then we escalate to a physics-based simulation tool like Rosetta.
Rosetta is checking the actual thermodynamic energy, right? Like, will these amino acids actually bind together in reality, or will the molecule just unravel the second it hits water?
Exactly. Rosetta calculates that thermodynamic stability. But the survey highlights a massive vulnerability here, the danger of gaming the evaluator.
Oh, this makes me think of a lazy, hyper-smart student. You give them a complex grading rubric, and instead of actually learning the material, they find a mathematical loophole in the rubric to score 100% without doing any of the actual work.
It's a perfect manifestation of Goodhart's law. When a measure becomes a target, it ceases to be a good measure. If you tell a generative model to optimize exclusively for a high Rosetta score, you won't necessarily build a good protein. It might discover a bizarre alien sequence of amino acids that chemically shouldn't exist, but mathematically tricks the Rosetta software into giving it a perfect score.
But in a real Petri dish, that alien sequence would just turn to mush.
Right. It generates sequences with high computational scores but completely unrealistic biophysical properties. So to stop the AI from cheating, the survey emphasizes multi-metric validation. The paper details a study by Johnson and colleagues where they abandoned single bouncers and built a 20-metric computational filter.
Wow, 20.
Yeah. By forcing the sequence to pass 20 different evolutionary, structural, and thermodynamic checks simultaneously, they boosted their actual real-world laboratory success rates by 50 to 150%.
To navigate that brutal 20-metric gauntlet without hitting those loopholes, the AI relies on search algorithms. Things like Bayesian optimization, which is this delicate mathematical balancing act between exploring and exploiting.
Bayesian optimization calculates risk. It builds a probability model to decide. Does the algorithm take a safe bet and tweak a known functional sequence region to get a slightly better result? Or does it take a massive mathematical leap into a totally unexplored, weird region of the sequence space for a potentially massive payoff?
And it uses Markov chain Monte Carlo, or MCMC, to keep from falling off a cliff while exploring, right?
Exactly. MCMC is a probabilistic algorithm that explores local branches of sequence variations. It carefully maps the surrounding mathematical terrain, checking structural constraints at every tiny step, ensuring the model doesn't wander off a cliff of instability where the protein would just collapse.
Let's pause and connect the dots. We have our generative architects. We've forced them to follow our structural and functional constraints. And we've guarded against hallucinations with 20-metric digital bouncers and risk-calculating search algorithms.
Right.
So the math works, but what happens when that math is synthesized in a wet lab and hits a real human immune system or a real industrial chemical? Let's look at where this is paying off today, starting small peptides.
Peptides are short protein chains, usually just 2 to 50 amino acids long.
Mm-hmm.
Right now, researchers are utilizing them to combat global bacterial resistance. Traditional antibiotics are failing because bacteria evolve too fast. So we use models like AMP Designer and AMP-JAN to generate antimicrobial peptides.
These are basically tiny biological missiles designed to puncture bacterial cell walls. But they have to walk a terrifying tightrope, don't they?
They do. The AI has to balance two directly competing chemical constraints simultaneously. The peptide must maximize its microbe killing power while maintaining near-zero toxicity to human red blood cells.
And if we scale up from tiny peptides, we hit the heavy artillery antibodies.
Antibodies are how we neutralize complex, shifting pathogens like viruses or cancer cells. If we connect this to the bigger picture, the survey points to the OAS database, which contains over 2.4 billion unpaired antibody sequences. It is a staggering repository of human immunity. AI models like Lambo2 and AGLM study this massive dataset to design specific regions of the antibody called CDRs, complementarity determining regions.
The CDR is the actual grabbing hand of the antibody, right? The part that reaches out and physically latches onto the virus.
Correct. By using AI to intelligently design just those specific grabbing hands, scientists can rapidly prototype antibodies to neutralize novel pathogens, exactly like they did with SARS-CoV-2. We are talking about accelerating therapeutic development by orders of magnitude.
Scaling up even further, we get to enzymes, the industrial workhorses. We mentioned Zyme-CTRL using text prompts to build them earlier, but there's a real-world application here that feels entirely like science fiction. A model called MUT-compute was used to optimize a peat hydrolase.
Right, PET is the plastic used in water bottles and packaging. Left to natural environmental forces, it takes centuries to degrade.
But this AI-optimized enzyme named FastPTase literally eats plastic. The AI optimized it to remain structurally stable and efficient at low temperatures and across wildly varying pH levels. That represents a timescale shift from 500 years of environmental degradation to a few days of biochemical breakdown. It industrial industrial-scale plastic recycling biochemically viable today.
It's revolutionary.
So what does this all mean? I mean, we are fighting superbugs, we are rapidly neutralizing viruses, and we have enzymes dissolving plastic waste in days. If this is also proving, why isn't every disease cured? Why isn't every industrial process perfectly green right now?
Well, because despite the breakneck speed of the algorithms, we are slamming into two massive roadblocks. The first is data scarcity.
Wait, you just said the OAS database has 2.4 billion sequences? How can we possibly be starving for data?
Because we have billions of raw sequences, which is just the alphabet. What we lack is high-quality, assay-labeled data. An assay is a physical laboratory test. We might know the sequence of letters, but we don't have the experimental evidence detailing exactly what that specific sequence does. Its exact folding stability, its binding affinity, its kinetic rate.
Oh, I see.
Raw data is cheap and abundant. Generating ground truth, physically verified data is incredibly slow and expensive.
And without that ground truth data, the AI is effectively guessing in the dark when predicting real-world performance.
Exactly. Which feeds directly into the second hurdle, the black box problem. Deep learning models are notoriously uninterpretable. If a generative model designs a protein, it passes all 20 digital bouncers, but then inexplicably turns to mush when we brew it in the physical lab.
The AI cannot tell us why it made the choices it did.
Right. It can't explain its own biochemical reasoning, which means we can't learn from the mistake. We lack the mechanistic understanding of the AI's internal logic. Bridging that interpretability gap is critical.
So, the authors of the survey state quite clearly that what biology desperately needs right now is an ImageNet moment.
A decade ago, computer vision was revolutionized almost overnight because researchers released ImageNet, a massive, standardized, highly curated data set of millions of labeled photographs. It gave every AI researcher on Earth a common, high-quality benchmark to train against.
Biology needs its own ImageNet. A centralized, rigorously labeled, community-standard data set of protein structures, functions, and physical properties. If we build that, we give these generative models the ground truth they need to operate at their full potential without hallucinating.
That is the ultimate bottleneck. The search algorithms are ready. The physical wet lab synthesis is getting cheaper. But the high-quality data pipeline requires a massive, unified, global effort.
We've covered a staggering amount of ground today. From the left-to-right limitations of autoregressive models to the noise-canceling brilliance of diffusion models seeing 3D space. We've seen how scientists are grabbing the steering wheel with inverse folding and text prompts, and how 20-metric digital bouncers are preventing the AI from gaming the laws of thermodynamics.
It's a lot to take in, and this raises an important question, something for you to mull over long after this deep dive ends. Right now, every model we have discussed is trained exclusively on Earth's evolutionary history. The billions of sequences we feed the AI are solutions that natural selection found over billions of years.
We are training it on the biology that already exists.
But as these non-autoregressive models become more capable of imagining beyond their training data, will AI eventually design entirely novel biological functions that evolution never even thought of?
Functions that were never required for survival on Earth, but might be required for, say, surviving the radiation on Mars, or cleaning up entirely synthetic chemicals that nature has never seen.
If AI can write a biology that evolution never dared to dream of, what kind of life does that build?
A completely synthetic branch on the tree of life, written by a machine. It's a heavy thought to leave you with. If you want to see the actual math and the specific algorithms making this possible, I highly invite you to explore the provided survey paper in our notes. We started this by saying that usually engineering is visible. You can't see these amino acids folding with your naked eye, but you will see the results in the medicine you take, in the climate you live in, and in the very fabric of life on Earth. The silent revolution is already here. Until next time.